Welcome! Today we're diving into a really fascinating medical puzzle called cryoglobulinemia. It's a condition where the very system designed to protect us, our immune system, can, well, become the source of the problem. Think of it like a loyal army that, when pushed too hard for too long, starts to rebel against the very kingdom it's supposed to defend. So, what does that very long name actually mean? Let's just break it down. Cryo means cold, and globulin is just a type of protein in our blood. So, pretty literally, it means cold proteins. These are specific antibodies that do something really strange when the temperature drops. They start to clump together, forming these precipitates that can clog up blood vessels and cause all kinds of trouble. So here's how we're going to tackle this. First, we'll get a handle on what these cryoglobulins really are. Then, we'll look at our normal immune army to see how things are supposed to work. From there, we'll see what happens when the system breaks, cover the 3 different types of the disease, figure out how it's diagnosed, and finally, look at how you treat it.
Okay, first things first. Before we can even begin to understand what goes wrong, we really have to appreciate just how elegant our immune system is when it's working perfectly. Let's just think of it as this highly trained, incredibly efficient internal army.
It's really a beautiful 4-step process. In step 1, an invader, an antigen like a virus, gets into the body. Then, step 2, our special forces, the IgG antibodies, move in. Their only job is to tag that threat for destruction. Once it's tagged, step 3, other soldiers like IgM antibodies and complement proteins pile on, forming this big, easy-to-spot clump. And finally, step 4, the cleanup crew arrives. Our macrophages just swoop in, swallow the whole package, and eliminate it. Threat gone. Clean, simple, and effective. Now, that system is brilliant for short-term fights. But what happens when the enemy isn't just a one-and-done invader? What if the threat is chronic? Like a persistent virus or an autoimmune disease that just never, ever goes away. Well, that's when this beautifully tuned system starts to crack under the pressure. And this right here, this is the million-dollar question. When the battle never ends, the whole game changes. The system gets pushed way beyond what it was designed for, and it starts to shift from a healthy defense into a state of chronic, damaging warfare against itself. So the army goes into complete overdrive. It's just constantly pumping out IgG antibodies to fight this thing. But this endless war starts to drain supplies. Those C3 and C4 complement proteins we mentioned, they're being used up so fast their levels start to plummet. The body gets so desperate, it even starts recycling old antibody parts to try and keep up. And then, in all of this chaos, a new, mysterious soldier shows up, something called rheumatoid factor. So, what on earth is rheumatoid factor? This is where things get really wild. It's essentially a case of friendly fire. It's an antibody that gets confused. Instead of attacking the enemy, it starts attacking our own IgG antibodies, the very soldiers it's supposed to be helping. And this is a massive and very dangerous turning point. Now, what's really fascinating here is that not all rheumatoid factor is actually bad. In a normal, short fight, our body makes what's called polyclonal RF. You can think of it as having a pretty weak grip. It actually helps in the cleanup process. But in a chronic, relentless battle, a single cell line can go rogue and just start mass-producing monoclonal RF. And this stuff has a super strong death grip on our own IgG. And that super strong grip, well, that's the whole problem right there. This monoclonal RF grabs onto so much IgG that it forms these massive, tangled balls of protein. They get so big that our macrophage cleanup crew just can't swallow them. They're basically indigestible. And these massive, uncleared clumps, these are the cryoglobulins. They are the villains of our story, now just left floating around in the bloodstream causing damage.
Okay, so now that we get how these rogue protein clumps form, it starts to make sense that this whole process can get kicked off in a few different ways. And that brings us to the 3 distinct types of cryoglobulinemia, each one with a totally different root cause. And this table here breaks down the differences perfectly. So look at type I. Notice how the rheumatoid factor activity is absent? In this case, the problem is with the antibody factory itself, usually a B-cell cancer, just overproducing one kind of antibody. But then look at type 2 and type 3, the mixed types. Here, rheumatoid factor is present. So the problem isn't the factory, it's a reaction to something else. For type 2, it's usually a chronic virus like hepatitis C, and for type 3, it's typically tied to an autoimmune disease like lupus.
So, with these 3 different causes, how in the world do doctors figure out which one a patient has? Honestly, it's like a detective story where they piece together clues from different lab tests to solve the mystery.
So the detective's first clue is actually pretty straightforward. They test for that key player we've been talking about this whole time, rheumatoid factor. Just finding out if it's there or not tells you a huge part of the story.
And here's how they read that clue. It's like a fork in the road. If rheumatoid factor is present, the detective knows the immune system is reacting to something. That points straight to type 2 or 3. But if the RF is absent, it means the problem isn't a reaction, it's a production issue. The antibody factory itself is broken, and that points right to type 1.
All right, the detective's next clue is to check the Army's supply levels. Remember those C3 and C4 proteins, the ammunition for the fight? Well, in types 2 and 3, where there's that chronic battle going on, the levels of these proteins are going to be low. They're consumed. It's kind of like looking at a fuel gauge and seeing it's on empty. It just confirms the immune system is under constant heavy strain.
So the final piece of this diagnostic puzzle comes from a really cool test called protein electrophoresis. You can basically think of it as a high-tech sorting machine for proteins. It gives us this detailed picture of all the antibodies in the blood, so we can see if they all look the same and come from one source, or if they're a diverse mix from lots of different sources.
And the results are just visually unmistakable. If the test shows a single sharp spike like you see on the left, it means one rogue cell line is just mass-producing one identical antibody. That's a monoclonal gammopathy, and it's the smoking gun for type 1. But if the result is that broad, wide hill on the right, it means lots of different cells are making lots of different antibodies, and that points our detective straight toward type 2 or type 3.
So the case is cracked, the culprit has been identified, type 1, 2, or 3, and now the strategy for how to fight back becomes remarkably clear, and the philosophy behind it is what's most important. And this right here is probably the most important takeaway. You don't treat the protein clumps, that's just the symptom. You have to go after what's causing them in the first place. If it's type 2 from hep C, you treat the virus. If it's type 3 from lupus, you treat the autoimmune disease. And if it's type 1, you treat the underlying cancer. You have to go to the source and shut the problem down. Period.
You know, when you step back, this whole condition gives us a really profound look into the delicate and sometimes paradoxical nature of our own biology. It really makes you stop and ask a fundamental question. How is it possible that a system designed for our ultimate protection can become the very source of our danger? It's a powerful reminder of the incredible and sometimes fragile balance that's going on inside us all the time.